class! So today we will continue our lesson on chapter 1, which is enzymes. And hi class! Today we will continue our lesson. Hi class! So today we will continue our lesson on chapter 1, which is enzymes. And today we will cover 3 subtopics. The first one is inhibition. The second one is allosteric regulation, and the third one is classifications of enzymes. So the first one, inhibition. Previously, we already learned about the factor that can affecting the enzyme activity, and one of it is inhibitor. So what is inhibitor? Inhibitor is actually refers to certain chemicals or substance that selectively inhibit the actions of the specific enzyme. And bear in mind, for the enzyme structure do have two sides. The first one is the active site, where the substrate will bind with the enzyme, and this is where the catalysis reaction will occur. Meanwhile, another site is named as allosteric site. Okay, so for inhibition, the process of where the inhibitor inhibit the actions of the specific enzyme, there are two types of inhibition. The first one is competitive inhibition. The second one is non-competitive inhibition. If the inhibitor bind to the active site of the enzyme, that is known as in competitive inhibition. And if the inhibitor bind to the allosteric site of the enzyme, it is called as the non-competitive inhibition. And non-competitive inhibition can be divided into two types. The first one is reversible non-competitive inhibition, where the inhibitor will bind to the allosteric site of the enzyme via weak interaction. Meanwhile, for non-reversible, non-competitive inhibition, this will happen when the inhibitor bind to the allosteric site of the enzyme by a strong bond. Okay, and bear in mind that all competitive inhibition is a reversible process. Okay, is a reversible inhibition. So let's look one by one. The first one is competitive inhibition. Competitive inhibitor, okay, are chemical agents that sufficiently resemble the normal substrate from doing so. So meaning to say, the competitive inhibitor have quite similar shape with the normal substrate of the enzyme. Okay, and then the actual enzyme thus competes for the positions of the active site of the enzyme. If the concentration of the competitive inhibitor is higher than the concentrations of the actual substrate, therefore the inhibitor will bind to the active site of the enzyme, and catalysis does not occur because substrate does not bind with the enzyme. For an example, succinate acid is a actual substrate for a certain enzyme. Meanwhile, a malonic acid is the competitive inhibitor for the enzyme. Okay, competing with the succinate acid. So bear in mind, the competitive inhibitor always bind to the active site of the enzyme by weak bond. Therefore, it can detach from the active sites of the enzyme. Okay, it is reversible. This one, as you can see, This is succinate or succinic acid. This one is malonic acid or malonate. Okay, the shape is quite similar, inhibitor with the substrate. So if the malonate, okay, bind to the active site of the enzyme, no product will be formed because no catalysis reaction occur. However, if the actual substrate did bind to the active site of the enzyme, therefore the catalysis can occur and thus producing the product which is fumarate. The next one is non-competitive reversible inhibition. Non-competitive reversible inhibition, okay, occur when an inhibitor they will form a weak chemical bond with the enzyme and they bind with the enzyme at the allosteric site, okay, not at the active site. Therefore, they are they are indirectly, okay, inhibit the catalysis reaction to occur. How? By altering the shape of the enzyme. So when the non-competitive reversible inhibitor bind to the allosteric site of the enzyme, therefore the shape of the enzyme will change and thus change the active sites of the shape of the active site of the enzyme, thus making the enzyme unable to bind with the substrate. Okay, so since the active site is not blocked, the inhibitor is not directly competing with the substrate molecule. Okay, example of non-competitive reversible inhibition is cyanide, in which cyanide will bind to the allosteric site of an enzyme, thus altering the shape of the active site of the enzyme and blocking the active site to be ready for the substrate. Last but not least is that non-competitive irreversible inhibition. This type of inhibition occurs when the inhibitor they will bind to the allosteric site because it is non-competitive. Then they make a covalent bond with the enzyme. Therefore, it makes the process is irreversible because the inhibitor cannot detach from the enzyme. So, since the inhibitor cannot fall off from the enzyme because it binds permanently, so it will change the structure of the enzyme and make it ineffective for catalysis reaction. Okay, so by time the enzyme will undergo denaturation process. Examples of non-competitive irreversible inhibition is silver and also arsenic. Okay, so this is examples of how does it look like. Here is the active site where the substrate can normally bind to the active site of the enzyme. Here is the competitive inhibitor, in which competitive inhibitor will bind to the active sites of the enzyme, thus blocking the actual substrate to bind with the active site. Remember, competitive, there are two types. Only one type of competitive inhibitor, which is competitive reversible inhibitor. And another one is non-competitive inhibitor, where the non-competitive inhibitor only bind at the allosteric site of the enzyme, either by strong bond, irreversible, or weak bond, which is called as a reversible. This is another figure showing how is the inhibition occur. This is the normal reaction, and this one is referred to competitive inhibitor, similar. The substrate binding is blocked. For non-competitive inhibitor, okay, once it binds to the allosteric site, so it will change the shape of the active site and blocking the inhibitor to bind at the substrate to bind it at the active site. So next is allosteric regulation. So allosteric regulation occurs when a non-substrate molecule, they either bind with the enzyme or they modify the site other than the active site of the enzyme. So meaning to say, the allosteric regulator is a molecule that bind to the allosteric site of the enzyme, and they can change the shape of the enzyme. Okay, and the changes in the shape of the enzyme can result in either activation or inactivations of the enzyme. So let's see. For the first one, okay, as you can see over here, active site is the active site of the enzyme does not is similar with the shape of the substrate, and when the allosteric inhibitor bind with the allosteric site of the enzyme, thus it change the shape of the active site of the enzyme. Okay, therefore substrate cannot bind. This one is called as inactivation of the enzyme. Okay, so they inhibit the process. Another one is that the active site of the enzyme does not resemble the shape of the substrate. Okay, because it in a distort shape. So what will happen? The allosteric regulator, which we call as the activator, will bind to the allosteric site of the enzyme and thus change the shape or altering the shape of the active site and allow the substrate to bind with the active site. This one is called as activation of the enzyme. Okay, where some allosteric regulator activate the enzyme. Therefore, allosteric regulator, there are two types, either they are inhibitor or activator. Okay, so this one similar as what I mentioned earlier. The binding of the activator can also stabilize the shape of the enzyme so that the shape of the active site of the enzyme is ready for catalysis reaction. Meanwhile, the binding of the inhibitor at the allosteric site, they stabilize the inactive form of the enzyme, okay, therefore bring to the inactivations of the enzyme. Okay, so these molecules may be products and substrates of a metabolic pathway. So what does it mean by it may be become the product or the substrate? So let's see here. This one we will discuss further in the negative feedback regulation later. So for allosteric inhibition, the binding of the allosteric inhibitor to the allosteric site, okay, can switch off the enzyme reaction, okay, because the binding of the regulator is by weak bond, therefore the process is reversible. So as you can see here, if the inhibitor bind to the active site of the enzyme, allosteric site to the enzyme will become inactivated. Okay, so if there is the allosteric site of the enzyme is empty because of the inhibitor detached, now the enzyme is ready for catalysis reaction. So now feedback inhibition. Okay, here, look at here, this figure here. This is substrate A. This is enzyme A. Substrate A bind to the active site of enzyme A, thus produce the B product. Okay, and then B product will become substrate for enzyme B, thus producing product C. And then the product C will become substrate for enzyme C, thus producing substrate product D, and etc. okay, and so on. And then last is that enzyme substrate E bind to enzyme F, thus producing the product F. And this of this product of this reaction can become the inhibitor for this reaction itself. Okay, when there is excess amount of product F that being produced, okay, the excess of product F will become inhibitor for enzyme A. Okay, it will bind to the allosteric site of enzyme A by weak interaction, thus inactivated the enzyme A. Once enzyme A is inactivated, meaning to say it's not active anymore, therefore we cannot produce product B, we cannot produce product C, D, E, and also F. Okay, so in conclusion, the end product are controlling their own rate of production, and there is no build-up intermediates of B, C, D, and E. Okay, this one also similar. Okay, and as I mentioned earlier, allosteric regulation or negative, other name is that allosteric feedback or negative feedback inhibition always involve more than one enzyme. Okay, this one is similar. Initial substrate is threonine, and then it will produce product. Product A will bind. It will produce product A. Product A goes to enzyme B, thus producing product B, and product B will become substrate to enzyme number 3, and so on, until they're producing the end product, which is isoleucine. So the excess amounts of the isoleucine will later bind to the allosteric site of the enzyme, thus, okay, change the shape of the active site, and it is no longer able to catalyze the threonine. Okay, so it inhibits its own mechanism. Last but not least is naming and classification of enzyme. Naming of enzyme usually random at the time of discovery. So if the enzyme is found during the digestion process, it is called as a digestive enzyme. Okay, an enzyme name also indicate both of the substrate that act upon the enzyme and also the type of the reaction that the enzyme catalyze. And another one is that by adding the word ASE, okay, at the root of the name of the substrate, such as urease. So meaning to say urease is enzyme for substrate of urea. Okay, the substrate is urea, the enzyme is called as urease. And enzyme can be classified based on the reaction, chemical reaction that they catalyze. And there are 6 classes of enzyme. We have hydrolase, lyase, isomerase, ligase, transferase, and oxidoreductase. So, what is oxidoreductase? Oxidoreductase is an enzyme that catalyzes the biological oxidation and reduction by they transferring either the hydrogen, they transferring the oxygen, okay, or electron from one molecule to another. Examples of oxidoreductase enzyme are dehydrogenase or oxidase. Cytochrome oxidase and also lactase dehydrogenase. Next is transferase. Okay, you remember naming the enzyme is based on its function, right? Based on its chemical reaction. So that's why it being called as transferase, because its function is to catalyze the transfer of functional group from one substance to another, from A to B. Okay, examples of enzyme transferase, transaminase kinase, alanine deaminase, and acetate kinase. Next is hydrolysis. Hydrolysis catalyzes the formations of two products from substrate by hydrolysis. Okay, by additions of water. That's why the enzyme is known as hydro. Hydro means water. Okay, hydrolysis. So enzyme examples are lipase, amylase, and sucrase. Next is lyase. Lyase is in contrast with hydrolase because they catalyze the non-hydrolytic addition. They does not involve water in the chemical reaction. Okay, and addition or removal of the group from the substrate. As you can see here, it causes the removal of the carbon dioxide. Example are decarboxylase, aldolase, and isocitrate lyase. Next is isomerase. Isomerase is the enzyme that catalyzes the internal rearrangement or isomerization. Okay, they only change the sequence of the functional group inside the substrate. Okay, this one is actually refer either the functional group or the atoms. Okay, rearrangements of atom within the molecules of this or the substrate. Example, glucose phosphate isomerase. Last but not least is ligase. Ligase is an enzyme that catalyzes the joining together of two molecules by simultaneously breakdown of ATP. Therefore, in chemical reaction that occur with the help of ligase always need energy, okay, to make sure that the process, the chemical reaction is occur. Examples of the enzyme is acetyl-CoA synthesis, phosphokinase, and also DNA ligase that you can found in the DNA replication process. Okay, so I think that is all for today, class. Thank you.